Hello everyone, I'm drberkkkaratas. We will be discussing exosome therapy for erectile dysfunction. Before we dive into the topic, let me say this. Since there is no clinical study yet, implementing this is not very appropriate right now since there are no studies on humans. There are preclinical studies and I will talk about those. It's a very promising treatment, a treatment that also eliminates some legal and judicial problems related to the root canal. So I think it will be quite successful and we will be applying this very frequently in the future. For him already, I've made the video, I'm making it. Yes, I just mentioned it. Basic and preclinical research is ongoing. It has been very successful. But since there are no clinical studies on humans, this means the following. When there are no studies on humans, how much dosage will you give in how many sessions and how will you prepare it? Since there is no template or guideline regarding these, it is not appropriate to implement this in the treatment of erectile dysfunction right now. So what exactly are these exosomes? What we call exosomes are actually small vesicles that are released from all the cells in our body. We can call them tiny vesicles. According to the cell, that they are released from. So it goes to the purpose for which it was released. So we can't say that exosomes have just one function, like it carries this, or it carries that. Whatever the releasing cell wants to do, it will communicate that to the receiving cell. Is it going to command healing and repair? Or is it going to command destruction? Or is it going to send a message? Accordingly, it carries something. It can carry DNA, it can carry RNA, it can carry a signaling cell, and it can send a signal aimed at healing. So actually, you can think of it as intercellular signaling. You can think of it like communication. It comes from the cell, it comes from every cell. They are very, very small. We can understand this like this. I animated this. I made an anime, I said, I did something, I adjusted their sizes. This is the diameter of an erythrocyte. So it's a red blood cell. The diameter of the cell that gives color to our blood. This is the exosome that it released. Look how small it is. They can release 1, 2, 100 or 50. These are, as I said, meant to serve a purpose. Now, obtaining these exosomes is quite a difficult procedure. I also had a hard time reading it. I had a really hard time understanding. Of course, since this is not part of my job, obtaining this is not my responsibility. Here, or repeatedly, the production of Santoff cells involves the use of some special kits, etc. It takes a bit longer for the culture medium, to be released from the cells and then to be collected. It's quite a lengthy procedure, but in the end, exosomes are obtained. Yes, this is the first study that has been done regarding this. Here, when he says rat, he means mice, in a rough sense, rats. It was done in 2017. What have they done with the mice here? They have created type 2 diabetes, meaning they made them diabetic. Then they gave the exosomes. And of course, they didn't just give the exosomes to these mice. At the same time, they also gave them stem cells and stuff. So normal fluid. They gave saline solution to have a control group. Here is something we pay a lot of attention to. Jar of pressure, arterial pressure ratio is there. Look, this is the value in normal rats or in normal mice. When diabetic mice are given exome, this is the value. So edit, this is only the value. When stem cells are made, when fat-derived stem cells are made, when normal standard saline fluid is given, the success rate of exome is almost as much as that of stem cells. So as we see here, if we had given exosomes instead of stem cells, we would have achieved roughly the same success. This is a very nice image. Look, it's a very normal, the image of the mouse, or meaning the image of the cavernosa inside the penis. Look at this red muscle. The red ones are smooth muscles and their numbers are quite high. When exosomes were administered, When stem cells derived from fat are given and saline solution is administered, you can see the healing here, almost with exosomes. The values are the same, or as when only stem cells are given. So he said that if you just give exosomes, almost the same healing will occur. This is the first study that was conducted. After this study, a lot of animal studies were conducted. Of course, we have two important studies that compile all these animal studies and produce results, which we call meta-analyses. Where do we obtain the stem things for these exosomes? From which cells are exosomes obtained? Exosomes can be obtained from samples taken from adipose tissue, bone marrow, and human urine. Really successful results were achieved. As I just explained, so there's not much difference between that study from 2017 and the other studies. I took it because it was the first one. So if it's the same as stem cells, it raises a question in people's minds. Of course, I also asked this first, if it's the same as stem cells, why are we making exosomes? We go through so many procedures. There are a lot of legal regulations regarding the application of stem cells. There are a lot of legal regulations because you are taking bone marrow from a person. The collection of the placenta is already subject to very strange rules. By the Ministry of Health, you're taking fat. These are always serious things, procedures, so legal and regulatory procedures. Exosomes are not quite like that because when you take it, you don't give stem cells, you're giving exosomes. So in a way, actually, from one person to another, it's like you're giving blood or platelets. So obtaining just exosomes, despite being quite cumbersome, legal is, and in medical dimensions, it's less. It seems like a procedure that requires a permit and has much more risks, but is easier to implement. Right now, of course, we don't know this very clearly because there are no human studies. So, what kind of problems might we face in the future? Or will we not face no follow-ups, but up to the router? We know that it is effective in preclinical studies. We will keep monitoring. As clinical studies come out, we will eventually be able to use them in our own clinics. I think we will apply it because it will emerge as a very promising procedure that is difficult to prepare, but very simple to implement. In the upcoming periods, that's all I have to say about exosomes for now. We are keeping track through scientific publications. And PubMed, if there are new developments regarding these and if they start to be implemented, we will also start applying these in our clinic here. Thank you.